So the first thing we're going to do is just a brief introduction to biochemistry and how it's common to virtually every living organism, whether it's an elephant or a bacterial cell or a duck. What goes on inside the cells is almost identical, right? There's a few species-specific and cell type-specific things that go on, but for the most part, the basics are all the same. And then we'll cover briefly a review of water, pH, and buffers, and how to do math without a calculator. Okay, so the first thing we want to do is, go through all that material again, is just introduce the concept of what is a cell, what's in it, how are the chemical components in a cell made up. So if you look at this little chart, this little table 1.1, and it lists what are the compositions of all the elements in human beings, seawater, and the Earth's crust. A very generic chart. So if you look at, let's do the seawater first. Right, so right in the center. Seawater is mostly what? It says roughly 2/3 hydrogen and 1/3 oxygen. Now we're not counting mass here, we're just counting number of atoms. So it's roughly 99% water, which makes perfect sense. It's seawater. And you know seawater is, we call it salt water or ocean water. There's some things dissolved in it, and the number one thing dissolved in it would be salt. So if you look down the chart, the next most abundant things are clearly sodium and chlorides, right? And they're roughly equal proportions. So that's mostly salt water right there. That's like 99.5% of the thing. And then there's some trace elements of nearly everything else, right? So carbon, calcium, magnesium, sulfur, and a few other things. Let's compare that to the Earth's crust. So the Earth's crust on the right is mostly composed of what? So look at the chart and tell me what you think. Anyone can answer this. Doesn't matter to me.
Oxygen.
So oxygen and
Silicon.
So what would that be? Like we said, when seawater was mostly hydrogen and oxygen in a 2-to-1 ratio, you're thinking that's water. What about the Earth's crust? It's almost half oxygen and about a quarter silicon. What would that be?
Silicon dioxide?
Silicon
It'll be
dioxide.
sand.
Sand, exactly. So it's mostly silicates or sand. So roughly, 70% or so of the Earth's crust we would call sand. Now that sand may be compressed into rocks, all right, like other more hardened things than you think of as just granular sand. But beyond sand, what's the next most abundant thing in the crust besides the oxygen and the silicon? What do you notice next?
Is that aluminum?
Aluminum, or aluminium if you'd like to pronounce it that way, or iron, calcium, right? So lots of metals in the crust, which makes sense, it's the Earth's crust, right? So lots of these other metals, again, mixed with oxygen would form lots of oxides or rust, right? So lots of oxides of metals as well. These are all your gemstones and ores and everything else we find on the crust, right? What is not in high abundance is carbon, right? It's only 0.19%. It's much more than in, say, seawater, but the carbon there is also locked up in things like graphite and other minerals. Okay? Now let's compare human beings. Us, we are most similar to which of those two, seawater or the crust?
Seawater.
By far, seawater. Look at the hydrogen and oxygen again. It's not quite 2-to-1, but it's mostly water again. So if you could describe us in the most basic terms, and this has been done in science fiction stories and everything else, where we're mostly water with a concentration of carbon. Right? That's pretty much what we are. So if you look at those, hydrogen, oxygen, and carbon together make up about 98% of all the atoms in you. Right? So where do we find the carbon? We said the hydrogen and oxygen, and clearly that's the water. Where do we find the carbon? What molecules make up all that carbon? So I can think of 3 good answers for that. Right?
Could you repeat the question?
Proteins, carbohydrates, and lipids.
Right. So the question was, what molecules contain that 9.5% of us that is carbon? And he said there's proteins, there's carbohydrates, and what was the last thing you said?
I said lipids.
Lipids. Okay. Is there any others? Proteins, carbohydrates, lipids?
Nucleic acids too.
Nucleic acids as well. And those 4 he just listed are the main 4 biochemical molecules we'll talk about. Okay. The next most abundant thing in that chart is nitrogen. Now if I include that nitrogen with the other three, now we're well over 99%, right? With everything else less than 1%. So where's this nitrogen going to be found? If you think
proteins, nucleic acids.
right, if you think carbohydrates, there's just carbon, hydrogen, oxygen. It doesn't have any nitrogens. Very few of them actually have nitrogen in them. So, like you said, they're going to be in the amino acids, which are part of proteins, and they're going to be in the bases of the nucleotides, which make up our DNA and RNA. Okay, so we are mostly seawater with the concentrations of carbon and nitrogen. That's a very drab look on human society, but we're more like seawater than we are the crust. Okay, so what do we need to know from organic chemistry? So we got to remember some of these compounds. So here's some organic compounds you should remember their structures and names for. So let's start at the left. We have alcohols, and if I take an alcohol and do some kind of chemistry to it, I could arrive at the molecules in the center, which are aldehydes and ketones. So from an alcohol to an aldehyde or ketone, what process would that be?
Oxidation.
Oxidation. So which I get, aldehyde versus ketone, would depend on whether the alcohol is primary or secondary. If the alcohol is tertiary, it can't be oxidized, right? And then if I take that aldehyde and oxidize it once more, I would arrive at the molecule on the right, the carboxylic acid. Okay, so these are different oxidation states of an alcohol. So we have to remember this from organic chemistry because we're going to use these functional groups quite often. On the bottom left there, it says, instead of an alcohol, it says a thiol or a sulfhydryl group, which is essentially the same structure as an alcohol, but the oxygen's been replaced by a sulfur, which is right below it in the periodic table. So do I need you to memorize the periodic table? Absolutely not. But you should know a part of it. You should know where hydrogen is. You should know where carbon, nitrogen, oxygen are, right? And you should recognize that sulfur is right below oxygen. It's a very small corner of the non-metals in the periodic table that you kind of remember from organic chemistry. And on the bottom right, we have our amines, which are either primary, secondary, or tertiary, depending on how many R groups are attached to the nitrogen. Okay, so these are your basic organic compounds. By basic, I don't mean acid basic, I mean elementary organic compounds that you need to remember in order to do this. Okay? On the next part are all the functional groups from those molecules we need to remember, and a couple more. Alcohols contain hydroxy groups. The next molecule on top says acyl. Keep in mind that does not read acetyl group, which would be a very specific acyl group, but acyl means the part of the carboxyl group that's not the O minus, right? It's everything else, the carbonyl on back. And the next one, of course, is just an isolated carbonyl, which is part of lots of compounds. The one on the right is our carboxylate, which is the conjugate base of a carboxylic acid. On the second row, we have, our sulfhydryl group, which is the functional group of a thiol. We have amino groups, right, whether they're protonated or deprotonated, right, with the NH2 or NH3+. And then on the bottom of it, we have a phosphate and a phosphoryl group. Now, you're probably familiar with phosphate. It's how lots of things are attached, and the phosphorus atom itself is never attached to anything but the oxygen. And the oxygen, of course, is attached to something else, possibly a carbon or nitrogen or something else. But the phosphoryl group on the right, where the phosphorus is directly connected to whatever chain it's on, carbon, nitrogen, something else, right? So which one of those do you think is a little more stable or a little less stable? We'll talk about those as we go through, right? So it makes a difference whether we're putting a phosphate on or a phosphoryl group. And I don't expect you to know that at the moment, but we'll get to there. And then at the bottom, when we start putting these things together, the first listed there says ester, right? And an ester is made from two molecules, right? Anybody remember what the two are? So,
An ether and a ketone?
Say it again?
An ether and a ketone?
It might look like that, but the ester is actually made from something. I put two pieces together and I lose an intermediary piece. I lose an intervening piece. So when I put
Carboxylic acid and alcohol.
Exactly. So if I put the carboxylic acid and alcohol together, I lose a water molecule in that condensation. So that it's not shown in the picture anymore. So it might look like there was an ether linkage and a ketone functional group there just kind of smushed together, but it's really a combination of an alcohol, which came from the carbon on the left, and a carboxyl group, which came from the carbon on the right, right? The real question is, the O in the center came from which of those groups?
Alcohol.
It came from the alcohol. Right? If you remember, the alcohol OH does a nucleophilic attack on the carbonyl. It's one of the most common reactions we're going to see. And the OH or O minus group that was on the carboxyl group left to form water. Okay? In the center, we have an ether group. What was that a combination of? Again, we lost water as a byproduct, and it was a condensation of what two things? And it's okay if you don't remember these. That's why I'm bringing these up.
Isn't it just 2 alcohols?
It is. It's 2 alcohols. The reason I'm bringing these up is so you go and review these things, so you know where they came from. We're going to see these types of linkages often. Okay, so again, a combination of 2 alcohols condensing and losing water makes an ether. The amide, or some people pronounce it amide, on the right is a condensation from what 2 groups? It's very similar to the ester.
Carboxylic acid and amino group.
Carboxylic acid, or a carboxyl group, and an amino group, exactly, or an amine. Okay? And again, we lost water in the center. Right? The bottom ones are a little more elaborate in that they contain this phosphor group. So we have a phosphate ester on the bottom left, or a phosphoester, and it's just like the ester above it, where instead of using a carboxylic acid and an alcohol, I used a phosphate and an alcohol. That's the only difference. And we're going to see this type of phosphate bond many, many times. The one in the center is again with a phosphate and a carboxylic acid this time. So instead of an alcohol, I've replaced it with a carboxyl group. Those come together and make a carboxyphosphate. And then lastly, if I use 2 phosphates, put them together, again, losing water in every one of these situations, I get a phosphoanhydride. And that one specifically states in the name it's a loss of water. It's an anhydride. And if you remember anything from organic lab and you're dealing with anhydrides, what did you have to make sure of your glassware before you began?
You had to make sure it was dry.
They're completely dry.
Completely dry, exactly. If there was any water in there, it would immediately break the bonds of that phosphoanhydride, or just any anhydride in general. Okay, so I'm categorizing these groups into 3 sets here. The ones in green, the ether and the amide, we're going to call very stable. That doesn't mean permanent. They are covalent bonds, all of them, but that doesn't mean permanent. They can be broken, it's just not easy, right? The ones on the left in purple, I'm saying they're easily broken, right? So hydrolysis using water or some other small molecule could easily break those bonds. And the ones in red are very unstable. And by unstable, I mean they don't like to stay in that arrangement, right? And those 2 in particular illustrate the very feature that makes them unstable. Does anybody know what it is already? And I'll give you a hint, it has nothing to do with those negative charges.
The amount of bonds?
So the amount of bonds. So look at your carboxyphosphate, and let's compare that to a phosphoester just to the left of it. Almost the same number of bonds, right? They're just arranged differently around that carbon. So it's not the number of bonds, it's how they're arranged. Right? So the carboxyphosphate and the phosphoanhydride have an arrangement of a pair of double bonds separated by 2 single bonds. Okay? And in both of those cases, we call that a beta-keto arrangement. We'll learn about these later, and beta-keto arrangements are considered very unstable. Okay? If they were separated only by 1 single bond, we would call those double bonds what? If it were double, single, double.
Conjugated?
Conjugated. And is that stable or not stable?
Stable.
That's a very stable arrangement. So we call those conjugated or alpha keto arrangements very stable. In this case, we have a beta keto arrangement. In other words, 2 double bonds separated by 2 single bonds. And one of them's got to be a double bond to an oxygen. But these are very unstable, and we're going to take advantage of that instability later when we look at reactions. Okay, so let's test your memory on a few of these things, and we don't have to have I'm not not picking on you, but let's try to let everyone get a chance to answer. This is to test everybody. What do you see in that first molecule? That molecule at the top left, labeled A, is one of the components to give beef its flavor, whether you like it or not. What functional groups do you see there? There's quite a few.
Alcohol.
There's definitely an alcohol, I agree.
Ether.
There's an ether. Can you be more specific? This sounds like Jeopardy now.
The bottom oxygen.
Yes.
The one that's, I guess,
not just a regular...
to the alcohol.
That's the correct oxygen, but not just a regular ether, but isn't it kind of attached to itself somehow?
Cyclic ether?
It's a cyclic ether, right? That makes a difference. And there's 2 more or 3 more groups you see in there.
Ketone.
Right, there's a carbonyl on the
Ketone.
left, right? In this case, it's a ketone. And what else do we see?
Alkene.
There's an alkene, right? A carbon-carbon double bond in there. And in fact, is this a stable arrangement with that carbonyl?
So conjugated.
Yes.
It's conjugated,
Yeah.
so it is stable. Good. And there's one other very, very minor functional group. Not really that functional. And it's off on the right. What is that?
Methyl.
It's just a methyl group, right? It's kind of the ignored child of functional groups, right? But it could make a difference in binding and other things, okay? Looking at the one on the right, choice B is a steroid hormone, testosterone. What functional groups do you see here? A lot of the same ones. Right, so you see the OH group, the hydroxy group on the top. There's a couple methyl groups drawn a little differently, but yes, they're still methyl groups. We have a carbonyl on the left, and again conjugated to another alkene, a double bond. The difference here is all these rings are not just bonded together, we call them fused, right? They're sharing a side. So this is a fused structure, which is very stable. Right, so this is one of your steroid hormones we're going to talk about when we get to lipids. Right? Part C is the vanillin, which is the flavoring agent in vanilla. What do you notice about this one? It's got quite a few groups on it.
Aldehyde.
There's an aldehyde. Which part's the aldehyde?
The CHO.
The CHO on the right, right. So this part's the aldehyde. And it's an abbreviation because you remember aldehyde was a carbonyl where the carbon of the carbonyl still had a hydrogen attached, at least one. And a way of abbreviating that is writing CHO. Okay, so there's an aldehyde on the right, and you've already pointed out the hydroxy on the left in several examples. What's the thing on the top?
That'd be
Ether.
ether methoxy group.
Exactly. It's an ether. That's its classification, and it's a methoxy in name. Right. And the thing in the middle, what's special about that?
Benzene.
Aromatic group.
Aromatic.
Right. It's a benzene ring. It's aromatic ring. All those things you said are correct. So is that a stable arrangement as well?
Yes.
Yes.
Stable.
Yes. So that's why vanilla tends to not fall apart in your cabinet. Right. It's a very stable molecule. Okay. Let's write them a little differently. How about D? What do you see in that one? So let's analyze the left side first. It says C4H9. How do you know the arrangement of that? Are there any rings in that? Are there any double bonds in that? How do you know?
There should be a CO double bond in the middle.
Right. Before we get to the CO, the first 4 carbons. You're right about the CO. We'll get there in a second. But the first 4 carbons, the C4H9, how do you know what that looks like? How am I confident that there's no rings?
Would you
Yeah.
count the degree of saturation?
I count degrees of unsaturation, right, or sites of unsaturation. And for 4 carbons, it should have how many hydrogens for a full complement?
12.
Not 12.
Oh, well, yeah, sorry. That would be all methyl groups.
Should
2
have
n
10.
plus 2.
Right, should have 10. It should fit the formula 2n plus 2, n being the number of carbons. So C4H9 is 1 shy of fitting the 2n plus 2 formula. And why is it 1 shy? Because it's connected to the next carbon, right? The one with the O on it. So I know the beginning part of it, it may not necessarily be 4 carbons in a row. Could be branched. But I know it's not in any ring structure, and I know it contains no multiple bonds. Okay? And then you told me the next carbon, correctly, was a carbonyl, C double bonded to O. How do I know that's not an ether? How do I know that it's not carbon bonded to O bonded to the next or last carbon?
We don't expect hydrogen on the carbon.
Right, the central carbon would have two missing bonds in that case. It would need two hydrogens to be that case. And it doesn't have them, so it must be a carbonyl. Okay. Good, so that overall, what kind of molecule is D?
Ketone.
It's a ketone, exactly. Okay, so this is all review, just remembering things from organic, and we'll see these structures over and over. E should be fairly simple. You see that C4H9 once again. That's just a regular old alkane sitting there with a CHO ending, which indicates aldehyde. Right, you guys did that one spectacularly in part C. And then finally, choice F here. Again, we begin with just an alkane at the beginning. What is this COOCH? It's a poorly spelled word, coach, but what does it really mean?
I think it's a carboxylic acid.
It's almost a carboxylic acid, and I'm not picking on you not picking on you at all. I get that answer every semester. I don't know why, but it's almost a carboxylic acid. Think a little farther.
Ester.
It's an
Ester.
ester. It's an ester, exactly. So if it ended in COOH, it would be a carboxylic acid, but it's got another carbon attached on that side. So it's been a condensation of the carboxylic acid you were seeing, which is there, plus a methyl group, right, or methanol. So we put those together to make this ester in part F. Okay, good. So you guys have already mentioned this part. There are four major classes. When I asked you where do you find all the carbons and nitrogens, and here's where they are. So we'll find them in proteins and nucleic acids, carbohydrates, and lipids. The first three of these have something in common that the fourth does not. Does anybody see a commonality in the first three that the fourth does not? There's more than one right answer here.
I mean, lipids are the only one that isn't polar.
Only one that isn't polar. Okay, that's true. Lipids are generally non-polar, not soluble in water. True. That's the other three can be very polar and very soluble. Doesn't mean they always are, but that's true. One other major difference. Nothing wrong with that answer, but I'm thinking of one other major difference too.
The number of monomers that make them up.
So the fact that they can be polymerized. And you said monomers, which means they can be put together to make multimers, right? So proteins are made of amino acids. Nucleic acids are polymers of nucleotides or deoxynucleotides. And some longer carbohydrates are made up of smaller carbohydrates, right? They can form longer chains. Lipids do not do this, right? So lipids do not form polymers. The other three do. Because that's the other answer. Okay? And that's known to show up on a test or two. So let's talk about the first one, proteins. So proteins, you probably learned this ages ago, are made of amino acids. And here's a typical amino acid shown on the right. It has a central carbon. We're going to call that the alpha carbon because the highest priority carbon in that group, if you remember back from organic, is the carboxyl group. That's the red one on top. So that's number 2 carbon, or the alpha carbon in black. And connected to it is an amino group. So these are known as alpha amino groups or 2 amino groups. And then it has a hydrogen and some other group called R, right? And R varies among the 20 different amino acids. So they all have that generic form, right? And then R can vary among the 20. R can be as simple as another hydrogen, and it can be as complex as a tryptophan, right? As the indole ring on a tryptophan. So it can get quite drastic, the difference between these two. Do you think that individual amino acid over there, whatever the R group might be, could interact in some way with another amino acid like it? It may or may not have the same R group, but it definitely has the same 2 carboxyl and amino functional groups. Do you think they could interact in any way?
Yes.
So what reaction could I do between one amino acid and another with those functional groups? We've seen this earlier.
Dehydration.
Right, the dehydration or condensation between the two. I can take the carboxy group of one and the amino group of the next and do a reaction. I lose water between the two, condense them together, and I make what type of bond? And don't say peptide bond.
Also the ester bond.
Say again?
Also the ester bond.
So an ester would be made between a carboxylic acid and an alcohol, right? So what would it be if it's instead a carboxylic acid and an amine?
An amide?
Amide or amide bond, right? However you'd like to say it. Right, so yes, so if you look at the very bottom, you can see there's two amino acids have come together to do just that. We have the carboxyl group from the first amino acid, right? And then the amino group from the second, forming that new bond in between, and it's an amide linkage, an amide bond. Now, if that's between amino acids, we give it a special name and call it a peptide bond, because it's between the amino acids of a polypeptide, or a protein. Okay. We'll go into proteins and amino acids much more when we get to that chapter, but just want to introduce all of these things to you in one slide set today. So the next one would be the other major source of all the, or major harborer of all our nitrogen, which is going to be our nucleic acids, made mainly RNA and DNA. So if I asked you RNA and DNA, what are they made of? And you could say, well, ribonucleic acid or deoxyribonucleic acid. What's the difference in those two things? So the main difference between RNA and DNA, there's two answers to that. So the two major structural differences between these molecules, RNA and DNA, are what? There's two major answers to this. And it's
An alcohol that's missing.
The alcohol group that's missing from where?
One of the ribose sugars.
Right, so on the right there you see a ribose sugar, and there's a highlighted hydroxy group on number 2. Right, so the carbon on the right would be number 1, that's where the base would be attached. And then here we have 2, 3, 4, and 5 sticking off the ring. But on 2, there's an OH highlighted. In the case of RNA, it's present. In the case of DNA, that's just the hydrogen there. That OH has been removed. Right, and we'll get to how that's removed in one of our last lectures of the semester. So it's either ribose or deoxyribose, and that gives DNA its name. That's one major difference. What's the other major structural difference?
Different bases?
Exactly. You see the bases at the bottom? There's the 5 of those shown. And a subset of those 5, 4 of them, will be found in RNA. And a different subset of 4 will be found in DNA. Adenine, cytosine, and guanine are all in common between them. And RNA will have uracil, where DNA will have thymine. And the only difference between uracil and thymine is a methyl group. Okay, so that's the two structural differences between RNA and DNA. All right. The other answer I get to that sometimes, which I'm thrilled you guys didn't say, is a lot of times they'll say RNA is single-stranded, DNA is double-stranded, which is not an absolute. RNA can absolutely be double-stranded. A lot of viruses have double strands of RNA. It can loop on itself and be double helix form. DNA can also be single-stranded, right? It's that way when it's being copied, right? It forms single or double-stranded depending on the conditions and what it can pair or not pair with. So don't say strandedness as your answer, because both could be both. All right, the answers to their structural differences are the missing or present hydroxy on the ribose and the subset of bases they use in the list below. Another common thing I see, and this is mainly, I know most, like 2/3 of you are biology majors, but this is the main thing we see in biology classes. And yes, I'll pick on biology occasionally. I'll pick on every class occasionally. But at the top, you see ATP drawn in the typical biology fashion as a ball of sharp energy for some reason. ATP is not a ball of energy. It's an actual molecule. And so we'll look at it in that form. ATP is an actual substrate, and it's used to make RNA. It's one of the 4 pieces you use to make an RNA molecule, the others being the CTP, the GTP, and the UTP. Right? And you see it's made of a ribose in black in the center. It's got an adenine on the right, and then it's got 3 phosphates on the 5th carbon on the left. Okay? So we'll think of ATP as this molecule, not some esoteric ball of energy. Okay? So how do you tell these 5 bases apart? Now, I know this semester is a little different. You guys are going to have all your materials during the test. You could pull up this slide if you wanted to, but if you learn them, it's so much faster. Right? So how can you tell these 5 bases apart? You may not be able to draw them straight out, but if I showed you a picture of one, you should be able to pick it out of this lineup instantly. Okay? So how do we do it so quickly? Can you give me a dichotomy between the two? Can you put two of them in one category and three in another rather quickly?
Purines and pyrimidines.
Right, so explain that a little more. Purines and pyrimidines, what's the difference?
Single or double ring structures.
Right, so purines being which one?
Double ring.
Right.
Double.
Okay, so who qualifies? We have adenine and guanine with each having two fused rings. So we're going to throw A and G into the purine bucket. That leaves cytosine, thymine, and uracil as the pyrimidines. Okay, good. We've separated them so far. If I give you one of these pictures and you have to decide which one it is, you already only have one of two or one of three choices. How can you tell A from G then? What's the glaring difference?
Carboxyl and a amine group.
Not necessarily carboxyl, but carbonyl. Right? So there's an oxygen in a G, and adenine has absolutely no oxygen. So if you look at the structure of adenine there, it's simply 5 hydrogen cyanide molecules put together. 5 H's, 5 carbons, 5 nitrogens. That's all it is. Right? And it can be made from that, actually. So if you want to tell A from G, just look for oxygen. A doesn't have one, G has exactly one. The other question is, how do you tell the pyrimidines apart? How do we tell C from T from U? Well, let's play the same game. How do I tell C from either T or U? Look at the oxygens. C has a single oxygen. Both T and U have 2 oxygens. So we can put these really quickly into categories. If it has no oxygen, purine, A. If it has 1 oxygen, purine, G. If it has 1 oxygen, pyrimidine, C. If it has 2 oxygens, pyrimidine, it's either T or U. Well, that leaves us with one question. How do we tell T from U? And if you look closely, thymine is just uracil with an added methyl group. And if you tried to write the word methyl using the letters from thymine, you're almost there. Right? It almost looks like the word. But in no way does the letters from uracil spell methyl. Right? So it's an easy way to remember which one is which. Thymine has the methyl, uracil does not. The last thing is, how do you know which one pairs with what? Right? You guys remember that C pairs with G and A pairs with T in DNA? And in RNA, we still have a CG pair and we have an AU pair. In every single one of those pairings, CG, AT, or AU, the combined number of oxygens is 2. So if you don't remember which one pairs with which, know that every pairing will have exactly 2 oxygens. So an AT pair, A has none, T has 2. And a GC pair, they each have 1. So the number of pairings, however you want to put them together, will always end up with a sum of 2 oxygens. With that very few rules, right, and that little bit of information, you can pick these 5 out of a lineup instantly. Right, let's move on to carbohydrates real quick. We'll just do a brief overview of carbohydrates. The name comes from the fact that they have carbon, hydrogen, and oxygen. And the formula looks like for every carbon, there's 2 hydrogens and an oxygen. So historically, they thought there's a water molecule attached to every carbon. So it's a hydrate of carbon, or carbohydrate. Right? We can put these together in multiple forms. They can have any number of carbons, really, but we'll stick to the common ones, which only have 3, 4, 5, 6, maybe 7 carbons. Right? And then we'll learn a few of these later, but one thing you got to get out of carbohydrates, and we'll cover it again at the same time, is that these molecules, like a lot of others, have a high degree of chirality. Right? There's going to be a lot of chiral centers. I know that was your least favorite word from organic when you learned chirality, and your second least favorite word was isomer. Right? Correct me if I'm wrong. So here, we're going to look at lots of isomers of sugars with lots of chiral centers. So it's the worst of organic chemistry that you remember put together. Right? So we'll see how these can be put together in long polymers, the difference between glucose and galactose, and monosaccharides and disaccharides, and so forth. Right? And then the molecule on the bottom right is a huge chain of sugars. Each one of those circles represents a sugar molecule of glucose, and that would be how you store it as glycogen in your liver and muscle cells. Lastly, we have lipids, and like you told me before, these are quite different from the others. They don't form polymers, and they're also not water-soluble, or they're non-polar. Okay, so some examples of those are waxes, like in the center molecule. We have triacylglycerols on the right, sometimes called triglycerides. And we have some steroids pictured in the center. Some other signaling molecules made from fatty acids like prostaglandins on the right. And we'll look at how a few of these are made. And at the bottom we have a typical cartoon drawing of these things showing a double membrane there, or a double bilayer of a membrane with the ball and the two green tails. Okay. It really doesn't look like that. This is another simplified drawing to give you the idea of the two leaflets. But in reality, it's that triacylglycerol you see at the top right, but instead of triacylglycerol, it'll be a diacylglycerol, only two tails attached, and one of them will be replaced with something more polar, like a phosphate or a sugar. Okay, and we'll take a look at all these when we get there later. Okay. So really quick, let's test you on these things. No points involved here, just test yourself. If something doesn't click and look familiar, remind yourself you should go review this. So in A, what does that look like? You can be as specific as you'd like, but all I'm looking for is protein, nucleic acid, carbohydrate, or lipid.
Phospholipid?
Right.
Lipid.
Absolutely, it's a lipid, and you're absolutely correct in it being a phospholipid. So that's what I was just describing as in the membrane. Okay. Now, B is not a trick question. I know it gives you the answer, but can you tell that B is part of a protein, other than the fact that it says protein? Right. Look at the structure and how they're linked together. You see lots of nitrogen. Right. You see lots of nitrogens, and they have a pattern to it where it's a carbon, and then a carbonyl, and then a nitrogen, and then a carbonyl, a nitrogen. It just repeats over and over. And there's a bunch of R groups that could vary. So yes, this is a series of amino acids in a polypeptide chain. Okay. How about C?
Carbohydrate.
It is a carbohydrate, right? In fact, those are a bunch of glucose molecules put together with a very specific linkage. You notice it says something like alpha 1,4 glycosidic bond. We'll learn what that means later, right? And there could be other types of bonds, right? And D,
Lipid.
it is a lipid. And more specifically, it's a what? You're right, it's a lipid, but more specifically, what's its name? Look at the functional group. That's a carboxylic acid, correct? But the other end of the molecule is a long chain of just hydrocarbons.
Just a fatty acid.
That's a fatty acid, exactly. So fatty from the fact that it's not soluble in water, it's got a long hydrocarbon tail. And then acid from the fact that it's a carboxylic acid. Okay. How about E? I know these are easy.
Nucleic acid.
Yes, that's a nucleic acid. More specifically, which one?
RNA.
And what gave it away? Two things.
The uracil and the alcohol.
Right, so everybody picks out the uracil right away. Had it not contained a uracil, you could still figure it out because the 2' still has an OH. Okay, good. How about F?
Steroid.
That is a steroid, which is a lipid. Exactly. And then how about G?
Protein.
Right, that's a protein. That's shown as a ribbon drawing from a crystallography structure or an NMR image or something like that. So we'll see them represented that way and how that's done. We'll talk about it, especially if you're in lab, we'll do that. Okay. All right, on to what these things do, not just, we listed the actors on the stage, now we got to tell you what their parts are. So we have DNA, RNA, and protein. And putting those together and how they communicate with each other and tell each other how to assemble, it was the central dogma of DNA. And so we have 3 processes here shown in arrows. It's actually a 4th process not shown by an arrow, which we'll talk about first. And what if DNA, this arrow is not on the screen, what if DNA could make a copy of itself, like an arrow from the DNA going back to the DNA? What would you call that process?
Replication.
Exactly. So DNA making a copy of itself is replication. We'll talk about that in a few minutes at the bottom of the screen. DNA with the arrow going to RNA is what?
Transcription.
Transcription. RNA with the arrow going to protein would be what?
Translation.
Translation. And RNA going back to DNA, that reverse arrow from right to left, is what?
Transcriptase.
So if DNA to RNA is transcription, RNA to DNA is
Reverse transcriptase.
reverse transcription, exactly. So one other question about these four processes, and I promise I'm not making fun of you. I do this with the class every semester, and I just want to see the difference. The DNA looping back to make another copy of itself, we call that DNA replication, is done by what enzyme? Yes, I know it's on the screen.
DNA polymerase.
Exactly. I know I wrote it at the bottom of the screen. That's the gimme question. How about the enzyme that converts DNA into RNA in transcription?
RNA polymerase.
Exactly, RNA polymerase. And the enzyme that turns RNA into protein in translation?
Ribosome?
Exactly. And every time I ask this question to the class, except for you guys, so congratulations, everyone has trouble with that last question. I don't know why. If I ask high school students, they get it right away. It's a ribosome. But when you ask college students, they forget that ribosome is actually an enzyme. Okay? And then the last one, RNA back to DNA, is what enzyme? Humans do not have this enzyme. Most organisms do not. It's very specific to some viruses.
Reverse transcriptase.
Reverse transcriptase, exactly. Right, so we don't make that enzyme. So which particular viruses would do that? It seems like we're going backwards.
Influenza?
Retroviruses?
Retroviruses, which doesn't mean they were made in the 70s. It means they're going reverse direction from the central dogma.
Like HIV.
HIV is an example of an RNA virus that does that, exactly. There are many examples of that, right? And some of you may be taking that class with me in the fall. So let's go over the processes real quick. DNA replication. DNA is copying itself, making new strands. It happens in the nucleus of a cell. Of course, if we're talking about prokaryotes, it just happens in the cytoplasm because they don't have a nucleus. And like you told me, it's catalyzed by an enzyme called DNA polymerase. Okay. Transcription, you told me, was catalyzed by RNA polymerase. It again uses a DNA as a template to instruct the polymerase what nucleotide to put in next for the growing RNA chain. Okay. All RNA is made this way. The most common one people think about is messenger RNA, but in fact, all RNA is made this way. Every RNA you can imagine is made this way, made by RNA polymerase. And this will come up later in the class, but RNA polymerase is a protein. Right? A protein is the machine that makes RNA. Right? And in the bottom, we have translation. RNA being turned into protein is done by the ribosome. And of course, most of the ribosome, like 2/3 of the ribosome in most species, is made of rRNA. And there's some helper proteins along the way that make it more efficient, but it could do it without them. And in some species, the ribosome is mostly RNA and very little protein. And in others, it's a lot of protein and very little RNA. There's a whole spectrum. But the key here is making proteins requires RNA machines. Well, I just said in transcription that making RNA requires a protein machine. So we have a chicken and the egg problem, right? So this is one of the major arguments against that RNA world hypothesis, because there is no way to make RNA without protein. Okay, so this is one of the major arguments against that RNA world hypothesis, and one of the arguments, or the main argument for RNA and protein or amino acids evolving simultaneously. Okay. All right, and then I have a set of slides here with this blue gradient background that I'm not going to go over with you on this video, but I want you to look over these and make sure you understand all these things from cell biology, right? It's how cells are organized and so forth, and sizes of things. I don't want to read those to you. They're just there for you to review in the slides. Okay? Or if you're watching this later in the video, you're going to have to pause it really fast. Okay? So let's finish the next part really quickly on some Gen Chem stuff, and then we'll take a quick break. So we're going to talk about water mostly, and I'm going to talk about some bonds from molecules real quick. So water is pictured on the top right, All right, so oxygen bonded to two hydrogens. You know it has two lone pairs. The oxygen's electronic geometry is tetrahedral, and its molecular geometry is bent or angular, leaving those two lone pairs with a more electronegative side and the hydrogen's a more electropositive side, making it polar. I think that was like 2 lectures in Gen Chem just now. All right, you guys know this stuff. But looking at some other bonds there, I want to point out a few in the list. Okay, so of these bonds, some of them have these really large numbers and some are very small numbers. So if we look at your typical bond in a water molecule, an OH single bond, it's listed as 463 kilojoules per mole. That's its bond enthalpy. In other words, that's the strength of that bond. Right? The larger this number, the stronger this bond, the harder it is to break. Okay? So an OH bond in water with 463 kilojoules per mole, that's 463,000 joules per mole, it's a fairly strong bond. But look right below it. We have an OO bond. It's only 146 kilojoules per mole. Where do we find an OO bond like that? Think about it before you answer.
Peroxides.
Peroxides, right. This is not a double bond. This is a single bond. It's not an O double bonded to another O, like in molecular oxygen, like you're breathing. This is an OO single bond that's only found in places like peroxides. And you know, you keep peroxide, like hydrogen peroxide, in a dark bottle in a cool place in your bathroom because it doesn't take a lot to break that bond. Right? A little temperature, a little light, it'll break. Okay, so that's why that number is very weak, 146. So it's a fairly weak covalent bond. So why is the OH bond so strong? Well, if we remember the chart of electronegativities from Gen Chem, right, as you go across the table, there are trends in the periodic table. Go back and look at those. If two atoms have a vastly different electronegativity, the bond will be stronger. Right? So who is the most electronegative element there is?
Fluorine.
Fluorine. So if I bonded hydrogen to fluorine, do you expect that to be a very strong covalent bond or fairly weak?
Weak.
Strong.
Very strong, right? And look at the difference in the numbers, right? An HF bond there is 567 kilojoules per mole. That is exceedingly strong, right? Look at the other halogens. They start to decrease as you go down the periodic table, chlorine, bromine, iodine, and so forth. And of course, I know you remember that hydrochloric, bromic, and iodic acids are your strong acids, but HF was not in that category because that bond is too strong. That's the reason. What if you have an electronegativity difference that's not so big, right? Then the number is going to be smaller. And what if you have an electronegativity difference that is 0, like hydrogen to hydrogen or oxygen to oxygen? Shouldn't be as strong a bond, right? You notice those numbers are smaller here. Okay, so this chart should give you an idea of the average energy or strength of a covalent bond. Even though they vary quite a bit in this table, they're all in the hundreds of thousands of joules per mole, right? So hundreds of kilojoules per mole, sometimes just 100, sometimes up in the 500s, but in the hundreds of kilojoules. These are covalent bonds, very strong. There's also some other weak interactions that are not covalent. We call these weak, or not necessarily weak by standards of other things, but they're non-covalent, intermolecular forces. So we call them weak bonds. And they generally don't act alone. They act in large numbers. So never underestimate the strength of these bonds in large numbers. So the first one we'll talk about is how do we get interactions between molecules that are not covalent? Why would they interact at all? Well, in this case, our water molecule, for instance, has some polarity to it. I said there's two lone pairs. They have more electron density than the hydrogens do. In other words, the electrons spend more time on that side of the molecule than they do on the hydrogen side of the molecule. So there's a permanent dipole moment there, right? So one side has a partial positive, one side has a partial negative, right? So that gives it the polarity that we need to do all these other things. Okay, so could these things interact with each other? Could a water molecule interact with another water molecule? Sure. If one is partial negative will interact with another's partial positive, we can have some kind of attraction between the two. Positive attracts negative. And their two negative sides would repel one another, so they would reorient to form a more favorable arrangement. Okay, this is called hydrogen bonding. Okay, so we have hydrogen bonding between these two things, like you see on the right here. The top figure shows two water molecules hydrogen bonding. The second one's two ammonia molecules. We could have a water molecule and an ammonia molecule in the next two drawings. So in this case, we can have a network of these things hydrogen bonding with each other. And although each one alone is not very strong, when they form a large network, like you see in the picture in the center, they can be fairly strong. This gives rise to the high boiling point of things with these hydrogen bonds, like water. An incredibly small molecule has an enormous boiling point, right? Or alcohols, it provides them with their high boiling points because of this hydrogen bonding. It also gives rise to things like surface tension and other things. Right? Okay, you also remember we could draw these things the way I'm drawing them on the right. These are called Lewis structures or Lewis dot structures. And you should remember how to do these. If not, let's go through one really quick, a really easy one. Let's do number 5 on this list. It's the most common one people use to teach this. And we're going to draw carbon dioxide. So let's go through the rules step by step so we don't leave anything out of how to draw the Lewis structure of CO2. So what's rule number 1? Remind me. When you approach drawing a Lewis structure, what should be the first thing you need to know?
Count all the electrons.
Okay, we could count the total number of electrons. Let's do that. And what do you mean by electrons? Not all electrons, but
Valence electrons?
valence electrons. What does that valence mean? I know this is all review from Gen Chem. I just want to make sure we're all up to speed.
Electrons in the outermost shell of the atom.
Right, so the electrons in the outermost shell. So the most we can have is how many for the elements
8.
we're going to deal with? 8. Unless we're talking about hydrogen and helium, then it would be 2. Okay, so good. Let's look at CO2. The carbon brings how many valence electrons to this party?
4.
4. And each oxygen brings how many?
6.
6. So we have a 4, 6, and 6, giving us a grand total of how many electrons?
16.
16. Are there any additional electrons I need to be concerned with? And what do I mean by that? If this overall molecule had a charge, let's say CO2, for some reason, had a -1 charge, how many total electrons would there be?
17.
17.
17. I don't subtract 1. The -1 charge means there's one more negative thing, and electrons are negative, so there must be one more electron. If CO2 for some reason had a +1 charge, then there would only be 15 electrons. And most often you're going to find that the number of electrons is an even number because they like to travel in pairs, right? So CO2, you told me 16 valence electrons. Good. What's the other thing I need to know before I start?
Shape.
I need to know who's in the center, right? How are they bonded together? So it's pretty obvious here the carbon's going to be in the center, but for the most part you have to be told. So let's put the carbon in the center and draw a single bond to each oxygen. Geometry is irrelevant at this point. We'll talk about it at the end. Just draw single bonds in any direction to a pair of oxygens. No double bonds at all, just singles. So those bonds you just drew represent a usage of electrons, right? Each covalent bond you just drew used how many electrons?
4.
Each bond you drew used how many?
2 for 4
2.
total.
So I used a total of 4 for the bonds I've drawn so far, which is probably what you were saying. So I've used 4 of my 16 electrons. How many remain?
12.
12. Where do I put them next? What's the next rule?
To fill in the 8 rule limit.
I fill in the shells of which atoms next?
Carbon.
Not carbon.
Oxygens.
Not necessarily always oxygen either, but the extremities first. Right? I fill in the extremities or the more electronegative atoms first. So here that's the 2 ends of the chain, the oxygens. And we can fill out those with 3 lone pairs each because that gives them a total of 8. Remember, the bond between the carbon and oxygen is 2. So we have to keep that in our count of electrons around the oxygen. So we can put 3 lone pairs on each oxygen. And does that account for all of our electrons? Yes, because that's 12 more. So 6 lone pairs, 3 on each oxygen, accounts for 12 electrons. We've already used 4. We're up to 16. We've accounted for all our electrons. Now check, are all the atoms happy? Right? In other words, do they fill their shells? Of course the oxygens have. We just solved that puzzle. But the carbon only has 4 electrons around it. And we'd like to have 8. We can't add any more. What can I do?
Double bonds.
I can promote the lone pairs to double bonds. Exactly. That does not change the number around the oxygens, but it does change the number around the other bonding partner, in this case the carbon. So I do that from one side. Doesn't matter which one. They're symmetrical. Do that from one. Now the carbon has a single bonded oxygen with 3 lone pairs still, and a double bonded oxygen with 2 lone pairs on it. The carbon's up to 6. We're almost happy. What should I do next?
Another double bond.
Which one? I have 2 options. I can take a lone pair from the same one I just did, or from the other one. Well, when you're drawing Lewis structures, you do all options. So we're going to draw 2 different structures here, both valid. So I would draw one where I now have a carbon with 2 double bonds to oxygen, and I'll draw one where I have a single and a triple bond to oxygen. Both are valid Lewis structures, but they don't have the equal validity, right? They don't have equal strength. The one where you drew it with the 2 double bonds is definitely more stable than the one with the single and the triple. However, in both of those cases, what is the geometry of this molecule? It's the same in both.
Linear.
It's linear. The carbon has 2 Vesper groups, right? Each oxygen, no lone pairs. So therefore it has linear geometry. So which one does it exist in most of the time? It goes back and forth. These are resonance between the 2, right? But the double bonded ones are far more favorable than the other one. Okay, so go back and do those 1 through 4 as practice if you need some practice drawing those Lewis structures. Okay. Some other forces going on between proteins here, and we'll go through the rest of these rather quickly because it should be all review. But we have different types of bonds. We have hydrogen bonds shown here between the amino acids in the protein. We have some hydrophobic interactions shown in these shaded areas where there's really no bona fide bonds. It's just hydrophobic interactions. And why do those even happen? What are these van der Waals or London dispersion or hydrophobic interactions really? There's no true bond there. Why do they stay together?
Hydrogen bonding?
No, there's no hydrogen bonding because there's no OHs or NHs or FHs or anything. Just it's greasy hydrocarbons.
Oh.
So in these shaded regions on this figure, you see that all these hydrophobic amino acids are coming together, right? But why do they stay together? There's no bond. There's no true bond between them.
Because everything else is watery and they don't like water.
Right, they'd rather not interact with water because that would require the water to become organized, right? The water doesn't want to interact with them either because it's polar and they're not. So it's just like you, if you were, I like using analogies, this will be the first of many silly analogies. If you were faced with something you cannot stand, right? You were put in a room and it's surrounded by the thing you cannot stand, whatever that might be, right? Maybe it's asparagus, I don't know. Whatever it is, you can't be around it, right? You just detest it. You'd rather leave the room. And if you leave the room and it's none of that around, you're free to roam around whatever you want. You're not detested by anything. But if you're in the room with it, you kind of have to stay away from it or face it, or you're more organized, right? So if you're excluded from that room, or if a water molecule gets kicked out of this region, it has more freedom. So it's increase in entropy. That's the reason they stay together. It's more energetically favorable to exclude all the water and interact with each other very, very weakly than it is to have a water molecule be highly organized in there in the middle of all these hydrophobic things. So it's an increase in entropy of the water that drives this interaction. It's not a true bond. Some others we have here, some salt bridges between the amino acids buried in the center of the protein. We have some hydrogen bonding. And on the right at the bottom, we actually have a covalent bond, a disulfide bond between 2 cysteine amino acids. Okay. Right, and then I have a little cartoon here that you can look at whenever you feel like it, explaining electrostatic interactions. It basically says that a positive charge and a negative charge interact with each other because they are attracted. And it's not permanent. It's not as strong as a covalent bond. It can be pulled apart by water. And if it's pulled apart, the farther apart the charges get, the weaker the interaction. I'm not going to make you memorize all the physics equations, right? But if you look at the equation, you can see that it makes sense. The Q's in that equation represent the strength of the charge, The stronger the charge, +2, +3, +4, the stronger the interaction. And you notice the distance between them, r, at the bottom. The farther apart they are, the weaker it gets by the square of the distance. Okay, this looks very much like the law of gravitation as well. Okay, so this is the strongest of the non-covalent bonds. Okay. Hydrogen bonding, we said, was fairly weak, but it must occur between two electronegative atoms, O, N, or F usually, and one of them has to have an H, otherwise it can't hydrogen bond. The other in the pair must have a lone pair, and that's almost guaranteed for O, N, and F. And the distance between the two is roughly 2.5 to 3.5 angstroms. What is an angstrom? Does that make sense to you guys? Have you heard that before?
It's just a very small unit of distance, right?
Right, it's just a unit of distance. It's kind of a, not really arbitrary assignment, but it's 1 times 10 to the minus 10 meters, right? So it's 1/10 of a nanometer. A nanometer is 10 to the minus 9 meters. So why use such an odd measurement? By odd, I mean it doesn't follow the -3, -6, -9, -12, -15 system. It's because 1/10 of a nanometer, or 1 angstrom, is about the width of a hydrogen atom. So if I asked you to measure how far apart are these two atoms, you would not give me the answer in kilometers. It would be a ridiculously small number, and it could not imagine it in my head. Also, if I asked how tall were you, you would not give me the answer in like nanometers or millimeters, because you're going, why use such a weird measurement? Just give me the answer in meters or feet, because I can visualize it. So the reason we use angstroms is because if I ask how far apart are these two atoms, and I say they're 2.5 angstroms apart, you can in your head visualize I could fit 2.5 hydrogen atoms between these two. You can see it's a meter stick on that scale. So that's why we use it. So when I say something's 2 angstroms apart, you go, oh, it's 2 hydrogen atoms width between the two. Does that help why that weird measurement's used? Okay, so that's our hydrogen bonding. It's very, very weak. We're at 2 to 10 kilojoules per mole. Remember, our covalent bonds were in the hundreds of kilojoules. So this one's very weak. And lastly, we have our van der Waals, which is the exclusion of water thing. They're just sticking together to stick together. And their distance is a little longer than a hydrogen bond, about 3 to 5 angstroms, and very, very weak, roughly 500 joules per mole, or half a kilojoule per mole. So a fraction, a very tiny fraction of a covalent bond. Okay. All right, so let's practice a little bit. At the bottom here, I'm going to ask a few association questions, and let's see if we can put these things together. So what kind of interactions would take place in a container of pure ethanol? And you remember ethanol just has 2 carbons and an OH group on it. That's it. Right? So what kind of interactions could happen in a container of pure ethanol? And I don't mean with the walls of the container, I mean among the molecules.
Hydrogen bonding?
There could be hydrogen bonding, absolutely. Are there any physical consequences of that?
You'd have higher boiling point
For its,
because of the
you're right, for its size, an elevated boiling point compared to, say, propane, which would be about the same mass and no hydrogen bonding. Okay, what else?
Surface tension.
Yes, you could have some surface tension, but we know alcohols in general don't have a lot of surface tension. But yes, it could have some effect on that. All right, what about the second one, acetone? Right, you guys remember what acetone looks like? It's the simplest ketone, 3 carbons long with a carbonyl in the center. Same question. What kind of things could go on in a container of pure acetone? Could we have hydrogen bonding?
Yes.
Explain how we could have hydrogen bonding. Remember, the only thing in this container is acetone. There's no water. To have a hydrogen bond, what's required? I need an O, N, or F with a hydrogen on it, and then another O, N, or F with a lone pair. That's almost guaranteed. So do I have an O, N, or F with a hydrogen on it?
No.
No. The carbonyl does not have a hydrogen on the oxygen. All the hydrogens are on the carbons. So acetone cannot hydrogen bond amongst itself. So consequences.
Lower boiling point.
Right, a relatively low boiling point for its mass, right, compared to, say, a 3-carbon alcohol, right, such as propanol. How about number 3? If I mix acetone and water, is there going to be hydrogen bonding going on?
Yes.
Yes, absolutely, between the acetone and the water and water and water. But I can mix acetone at any concentration with water. And so you imagine if I put a little acetone in water versus a little water in acetone, which would be mostly acetone, which one of those do you think would have a higher boiling point? The mostly water or the mostly acetone?
Mostly water.
Mostly water, because it can hydrogen bond amongst itself, and it doesn't care if the acetone's there, regardless if acetone's around. But if acetone is the majority of the solution, it can only hydrogen bond with whatever water is present. So the number of bonding partners is very limited, right? So as I increase the amount of acetone in water, you'd expect the solution's boiling point to drop. Okay, good. And lastly, we have acetic acid. You guys remember what acetic acid looks like? It's, again, 2 carbons, but it's a carboxylic acid on the end, right? Could that hydrogen bond amongst themselves? Remember, the second or first carbon is a COOH. Second was just a methyl.
Yeah.
Yes, it can hydrogen bond amongst itself. And very specifically, if you take your hand, I usually do this demo in class. You can't see my hands at the moment, but if you take your hands and hold it up like you're going to make that picture frame with your fingers, right? One hand being one acetic acid molecule and one hand being the other, and how your fingertips are meeting to make the picture frame. That's how the 2 acetic acid molecules hydrogen bond. It's called bidentate, or 2 teeth. In other words, the carbonyl of one is hydrogen bonding with the OH of the other, and the carbonyl of the second is hydrogen bonding with the OH of the first. It's like they're holding on with both hands. Right, so they hold on very tightly, which would have a drastic increase in their boiling points. So carboxylic acids have exceedingly high boiling points for their mass, much more than alcohols.
If we were to say so, acetic acid undergoes some form of reaction where the alcohol of the carboxylic or of the acetic acid loses its hydrogen, would you experience electrostatic repulsion between those?
So let's say you take acetic acid in your scenario, and it were to deprotonate. That's what you're saying, right?
Yes.
So it does do that in solution. We can actually calculate how much would do it by its pKa and the temperature and so forth. But you know acetic acid is not a strong acid, so it won't completely dissociate. Could we get it to do that? Sure, let's add a ton of strong base and completely deprotonate it, right? And turn it completely into acetate. The acetate molecules could no longer hydrogen bond amongst themselves. That would be true. But it's very difficult to do that and not have any water around. Because the moment you add, say, sodium hydroxide to remove all that, you're making water, right? When you add acid and base, you make water and salt. So you're right. If you could get to do that, they would not want to stay together, such as powdered acetate or something. But it's very hard to do for a weak acid.
Okay.
But good question. And in our last one, I already mentioned this, with our entropy, if I have 2 nonpolar molecules here, it looks like little bees flying around them, but those are water molecules. And if you count it up, we know there's plenty more than just 7 water molecules around this thing. But in this scenario, let's say there's 7 around each molecule. If they were to approach one another and bond in this hydrophobic interaction, it's not a true bond, then the number of water molecules needed to maintain the shell around them would not be the sum, wouldn't be 14. It would only be, say, 9. And I have 5 molecules free to go, do whatever they want. Their increase in entropy is what drives this process. So I'm burying a surface between the two is what this is showing. These are very, very weak interactions. Okay. And then there's a few more slides here that I'm not going to read to you, but this folding of proteins, this hydrophobic effect, is what allows proteins to assemble and fold together. One of the two major ways they do it. And then finally, we have our discussion of acids and bases. This is a review from Gen Chem. And what I want you to get out of this, and I'm putting most of the slides in here for you to just look at and remember, is we have 7 strong acids. You probably remember them. And anything weaker than that, we call a weak acid. And anything called a strong base, like your, hydroxides and like CH3 minus or peroxides or superoxide or something like that. If I allow it to do what it's going to do and become the conjugate acid, or in the case of, say, hydrochloric acid, become the conjugate base, they become very negligible as bases. So, for instance, hydrochloric acid, very strong acid. It will deprotonate completely, forming chlorides. The chloride is a negligible base. It's a very terrible base, right? It's unlikely to grab a proton from someone. However, if I take a very negligible acid like methane, methane is a terrible acid, right? It doesn't want to lose its proton at all. But if I rip a proton off of it and make it CH3 minus, that is an incredibly strong base. So they're equivalently weak and strong on the opposite sides. Look at this chart and you'll see where water appears twice in this list. Once appearing as an acid, H3O+, and once appearing as a base, as OH-, right? So water can gain or lose 2 protons in this case, right? Or gain or lose 1 from water itself. Anything between the two in strength we call weak acids and bases, because in water, the water is a stronger acid or base. Does that make sense? Right? So if it's outside the two, we call them strong acids and bases. Between the two, weak. In other words, they don't deprotonate 100%. And we have a number to describe that. It was pKa. And you guys remember how to do the pKa formula. P simply means take the negative log of whatever it is. So if I have the Ka, or the association constant, and take the pKa, it's the negative log of that number. Same thing with concentrations. If I have the concentration of hydroxides or concentration of protons or hydronium ions, and I take the negative log of it, it is the pH or POH and so forth. Okay. Right. That's all these slides are reminding you how to do that simple math. The one number I do want you to remember, it'll make life so much easier for you, is the square root of 10. Okay, so the square root of 10 is about 3.16. So let me quickly go through these 5 problems with you, and you can do these without a calculator, I promise. Doing it on a calculator is slower. So if I asked you what is the proton concentration of a solution whose hydroxide concentration is 10 to the minus 8 molar. Well, you know the product of the two must be 10 to the minus 14 molar. And hydroxide being 10 to the minus 8, the proton concentration must then be what? If the product must be 10 to the minus 14, the exponents must add to minus 14.
10 to the minus 6.
It's 1 times 10 to the minus 6 molar. That one's very simple. Okay. So next question is very similar. If I give you the proton concentration or hydronium concentration, you can give me the hydroxide concentration. Number 3 switches it up a little bit. I don't give you a 1 in front of the power. So how do you figure it out? The product is still 10 to the minus 14. So this is not a trick with math, or I guess it is, but it's very simple addition and multiplication. If the hydroxide concentration is that number sitting there, 3.16 times 10 to the minus 2, the hydronium concentration must be a number that when you multiply it times that gives me 10 to the minus 14. I'm going to rewrite 10 to the minus 14 as 10 times 10 to the minus 15. Make sure you agree that is the same number, because I added a 10 in front and I lowered the power by 1. It's the same number. So now I can multiply in columns. 3.16 times what gives me 10? Well, that would also be 3.16 because it's the square root of 10. And 10 to the minus 2 times what gives me 10 to the minus 15? Just subtract. It's 10 to the minus 13. So the answer is 3.16 times 10 to the minus 13. Right? Let's say you didn't have 3.16. Let's do a different one. How about number 5 at the bottom? It's 4 times 10 to the minus 3 molar. The other number would simply be what? 4 times what number gives me 10? That's 2.5. And 10 to the minus 3 times what number gives me 10 to the minus 15? 10 to the minus 12. So the proton concentration, or hydronium concentration, is 2.5 times 10 to the minus 12. Way faster than you can type it in your calculator. And if you miss a zero or a button in your calculator, you're going to believe the answer. Okay? So go through these problems and make sure you can do them. And again, I have a set of slides here explaining that process of all the acids, bases, and pKa's at the end, okay, including our Henderson-Hasselbalch equation.